Good morning, student. नव नवयुवक शिक्षण प्रसारक मंडळ चापोली संजीवनी महाविद्यालय चापोली welcome you in e-learning platform prepared by myself Dr. Shubhangi Prabhakarao Chavan, Assistant Professor and Head Department of Botany. Now, Swami Ramanand Tirth Marathwada University Nanded Semester Pattern Curriculum under Choice Based Credit System CBCS for Undergraduate Program, Faculty of Science and Technology, Subject Botany, Year BSc Third Year, Paper Plant Pathology. Now, student, in last lecture, our studied on ticca disease of groundnut. Now, today we will discuss let blight disease of potato. Now, the disease causes blight symptom which appear towards middle to late in the season. That's why the name is given. Potato is a native of the North Indies in South America. In Italy, late blight of potato was available in the area as an endemic disease. Gradually by 1842-1847, it spread throughout USA and Europe. Now, the disease became well established in Ireland, Europe and England by 1842. Later in 1845, it spread as epiphytic disease throughout the Europe and proud famine in Ireland, causing the death of 1 million peoples and more than 1 million migrated to other countries. In India, the late blight was introduced in the Nilgiri Hills between 1870 to 1880 from England. Nowadays, the disease is frequently available in the potato growing areas of the India. Next, host Solanium tuberosum. Symptoms of late blight of potato are available on foliage and inside the tuber. First of all, on foliage, symptoms appear as hydrotic area with indefinite margin at the apex and edges of the leaflets. The infected region gradually turn brown to black due to necrosis. In the early stage of disease development, chlorotic border develops around the necrotic regions. During the moist weather, the disease progresses very rapidly, causes much more days of the infected regions and produce a characteristic bad odor. The sporangiophores and sporangia are visible as matty growth on the abaxial surface of the leaflet. The entire plant may be damaged completely within a few days. Now, next, the tuber, the pathogen does not go down through the stem to the tuber. The tuber infection takes place in different ways. First of all, during growing season, hydrotic areas develops on the tuber surface and the regions become necrotic. If condition favors, the entire potato turns brown and become damaged completely before harvest. Now next point, during harvest. During harvesting, the tuber with the delicate skin may get ruptured and the tuber may find contact with the infected leaf and causes infection. Later the symptoms is visible after cutting to tuber as a wheel marked by small brown dots. Now next, this is the diagrammatical representation of leaf blight of potato. These are the leaf, figure number A, blight leaf, and figure number B, this is tuber of potato. Now next, causal organisms. The causal organisms of Phytophthora infestans belong the subdivision Mastigomycotina. Under the division Eumycota, the plant body is conicyately mycelium which grows both intra and intracellular inside the host tissue. The sporangiophores develops either singly or in group on the young seedlings and also from the lower side of the leaf of outlet plant. Now it is branched and develops sporangia at the apice. Innately, one sporangium develops at the apex, and at maturity the sporangiophores elongates from one side and shifts the sporangium. Towards the opposite side, another sporangium develops at the apex. Now, the diagrammatic representation of leaf blight of potato. Figure number A, we can see section of diseased leaf showing the developing sporangiophores coming out through stomata. Now next, figure number B, we can see number of sporangiophores coming out through stomata from a diseased tissue. Now next, figure number C, sporangium on the branched sporangiophores. Now next, figure number D, we can see a single sporangium, and figure number E, the germination of sporangium by producing zoospores. Figure number F, direct germination of sporangium, and lastly figure number G, we can see single oospores. The diagrammatic representation of leaf blight of potato. Now next, the sporangia are lemon shaped and colorless with a small stalk below and thin papillae above. During the favorable conditions, sporangium produce a number of bifragilate secondary zoospores. After sowing for some times in develop or thin film of water, the zoospores insets and the germinate by producing germ tubes. The germ tubes penetrate directly through the stomata or develops epizorium at its tip contact with the cell wall, which then penetrates the host wall both by the mechanical pressure as well as by enzymatic action. During the unfavorable condition, the sporangium behavior as conidium which germinate directly by the producing germ tube. Now, next, optimum temperature for the growth of mycelium is 16 to 18 degrees centigrade for the spiralization in 81 degrees centigrade for the germination of sporangia by zoospore. In 12 degrees centigrade while at 21 degrees centigrade it germinates directly by germ tubes.